All right, in this lecture, we are going to talk about the G protein linked receptors. And I'm going to start off with an introduction, then go into the structure and function. I'll talk about the two most important pathways that you have to know, and then give you a couple of mnemonics, and then a couple of knowledge challenge questions, and that'll be that. Let's get started. So I'm sure that you've talked about the G protein linked receptors in school. However, you might not have called them G protein linked receptors. They go by other names like G protein linked second messengers or G protein coupled receptors. That's a pretty common one. Even conversationally, a lot of people just say the G proteins. So I'm going to break down those words a little bit later and tell you what each part of it means and why we use those names to refer to these proteins. But first off, what does the G stand for? Why is it G protein? Well, the G stands for guanine. These are guanine nucleotide binding proteins, meaning that they bind guanine nucleotides, like, for example, guanosine triphosphate, or its slightly shorter little brother, guanosine diphosphate. These receptors are extremely important physiologically. We use them all over our body, and they do all kinds of different functions. These receptors are only found in eukaryotes and are the target of about 40% of all the drugs we use in medicine today. So I just told you that they're very diverse in function. However, there's two main signal transduction pathways that you gotta know. You have to know them for your boards. And that is the cyclic AMP dependent pathway and the phosphatidyllinositol signal pathway. Okay? And I'll talk about those more in a little bit. First, let's jump into the structure and function of these things. It's pretty cool. So I told you that I was going to break down the names so that you can understand the concepts a little bit better. So first off, let's get started with what does the word G proteins mean? Well, the G proteins are a component of this receptor system. The G proteins are heterotrimeric GTP binding proteins made of 3 subunits, hence heterotrimeric. So first there's the alpha subunit, then the beta subunit, and then the gamma subunit. And you should also notice from my illustration here that the beta and gamma subunits are more tightly linked together than the two are to the alpha subunit. So actually the beta and the gamma stay connected where the alpha subunit separates itself in normal function. These G proteins live on the intracellular side of the cell membrane and are attached to or connected to the cell membrane itself by lipid anchors. So the next thing for us to talk about is to define what are the G protein coupled receptors. Well, these are receptors that live in the membrane, and in fact they pass through the membrane 7 times, which is an important number. You might be tested on it. Alright, so these G protein coupled receptors interact with the G proteins and cause oftentimes a conformational change so that the alpha subunit of the G protein can accept different nucleotides. What about the term G protein linked second messengers? Well, that's another component of this system, which are cellular target proteins or enzymes that perform some kind of function when they interact with the G proteins that we've already talked about. Now, they are very diverse, but as I said in the beginning, there's 2 main pathways or 2 main targets that cause a pathway that you have to understand. And those 2 are the cyclic AMP dependent pathway and the phosphatidyllinositol signal pathway, which I abbreviate here as IP3-DAG pathway. So right now you're probably thinking, Dr. Joel, I have no idea what you're talking about. Don't worry, I will show you the whole thing working, and we'll talk about the parts as we go. And I'm pretty sure that it'll make sense after we go through it. First, we have to set the stage a little bit. The G proteins, or more specifically the alpha subunit of the G proteins, is bound in its resting state to guanosine diphosphate, or GDP. In this state, the receptor is just sitting and waiting. On the extracellular side of the cell membrane, a ligand comes and it binds with the G protein coupled receptor, causing a conformational change. The conformational change allows the alpha subunit to bind with guanosine triphosphate and release the guanosine diphosphate. Now the G proteins are active, and as active G proteins, they do a couple of things. First off, the alpha subunit separates itself from the beta gamma complex. This kind of frees it up a little bit, so it's free to move around. It's still attached to the membrane by a lipid anchor, but the lipid anchor can kind of swim through the membrane, so it's able to move to the target. It moves to the target, and it causes the target protein or enzyme to change in some way. Either the G protein interaction causes the target to speed up what it's doing, or slow down what it's doing, or stop what it's doing. It does something to the target protein. So in this case, I'm going to say that this target enzyme gets activated by the alpha G protein here. And once it's activated, it produces a product. And that product goes off and does another job, so it becomes a second messenger. All right? So the next logical question would be, what stops this process? Obviously, it doesn't run on forever. Well, inside the cell, there's GTPases, and they come and they hydrolyze the GTP back into GDP, which deactivates the target enzyme and separates it from the alpha subunit, which allows the alpha subunit to go back to its friend, the beta gamma complex, and the G protein coupled receptor, resetting itself, and the process can now start all over. So in order to understand the pathways, let me first show you that there is a difference in some of the G proteins, specifically the alpha subunits. So depending on which pathway that we're talking about, we're actually using different G proteins. In the cyclic AMP-dependent pathway, which I'll use as my first example, we use two different kinds of G-alpha proteins. The first one is G-alpha sub S. And in conversation, most people just call this the GS, or the GS protein. It's stimulatory, so it increases the activity of this pathway. The next one on the cyclic AMP-dependent pathway is the G-alpha sub I, and we just call that GI most of the time. That particular G protein is inhibitory to this pathway. It decreases the activity. On the other side, with IP3-DAG, the G-alpha protein we're using there is G-alpha sub Q, or just GQ. Let's get into a little bit more detail on both of the pathways and how they work exactly, starting with the cyclic AMP pathway. So I've already told you that the GS is stimulatory and the GI is inhibitory, but the rest of the pathway is the same. So we start with our G protein coupled receptor as the whole unit. When that receptor is stimulated, we know that the alpha subunit exchanges GDP for GTP and then migrates the membrane to act on its target enzyme. So what target enzyme is that? Well, that's adenylyl cyclase. Does that make sense? What does that do? Well, this enzyme converts ATP into cyclic AMP, and then cyclic AMP goes on to activate protein kinase A, which is an important enzyme in several other metabolic pathways. And I'll show you a little bit more detail on protein kinase A in just one second. What about GI? What does the GI protein do? Well, it would also begin with a receptor. It would be slightly different, remember, because it would be a GI receptor, but it would just have an inhibitory effect on adenylyl cyclase, which would decrease the production of cyclic AMP and thus decrease the activation of protein kinase A. So you just need to know that one increases it and the other decreases it. It's easy. How are you going to remember this diagram? Well, I think of it as a gigantic D and an L. Why D and L? Because adenylyl has both a D and an L. Next in our discussion is the IP3-DAG pathway, or the IP3-DAG pathway, or you might hear it called the phosphatidyllinositol signal pathway. We go, what? That's a big name. Yeah, so let's just say IP3-DAG. The IP3-DAG pathway uses GQ protein coupled receptors, and the target enzyme is a membrane-bound enzyme called phospholipase C. What does phospholipase C do? Well, it is bound to the cell membrane, and it actually uses parts of the cell membrane to make its second messengers. So it takes some of those molecules, called PIP2, or phosphatidyllinositol 4,5-bisphosphate, and it breaks that molecule down into two parts. The first part is IP3 inositol 1,4,5-triphosphate. IP3 is released into the cytosol of the cell, and it goes along and activates receptors in the smooth endoplasmic reticulum, which opens calcium channels and thus liberating calcium. And we all know that calcium has a very strong role in changing the physiology in a lot of things in a cell. So that's one mechanism by which this receptor works. The second half is the DAG half, or diacylglycerol. Diacylglycerol remains in the cell membrane. It doesn't migrate through the cytosol like the IP3. However, now that its structure has been changed by the phospholipase C, it can interact with another enzyme called protein kinase C. And protein kinase C, for its part, activates other pathways by phosphorylating different cytosolic proteins. And I will show a little bit more detail on that in the next slide. But for now, how do I remember this? Well, I think of a fork, like a dinner fork, like what you would eat with, because that's just what it looks like to me. And now for a couple of real quick words on the IP3-DAG pathway, just so that you can see it. So as we've already talked about, our GQ protein comes and it activates phospholipase C. Phospholipase C takes PIP2, which is already in the membrane, and it cleaves it, making IP3 and DAG. IP3 is free to roam through the cytosol, where it eventually activates calcium channels on the endoplasmic reticulum. This releases calcium, and calcium has an effect on a lot of different things in the cell. DAG, on the other hand, it stays in the membrane because it still has those hydrophobic chains or tails, which keep it anchored to the membrane. DAG helps to activate protein kinase C, and protein kinase C can phosphorylate other proteins or substrates, and the signal process is perpetuated. Okay, cool. And now into the mnemonics about how you're going to remember all this stuff and vomit it back up on your tests. Okay, starting with the IP3-DAG pathway. You absolutely have to know that the GQ protein is associated with this pathway. What about all the receptors that use a GQ protein? This is also high yield. I like the mnemonic, and I used the mnemonic when I was in school, have one M&M, which is written out like this for the histamine receptors, the adrenergic receptors, the vasopressin receptors, and the M is for muscarinic, aka the cholinergic receptors. On the other side, the cyclic AMP2 G proteins, what are they? GI and GS. You got to know it. You have to know it. Very, very important. For the GI, you're going to remember MAD2s, okay? For the cholinergic, adrenergic, and dopamine 2 receptors, subtype 2s. For the GS, it's everything that's left over. Beta 1, beta 2, dopamine 1, histamine 2, and vasopressin 2. So, if you need to review what those receptors are or what they do, here are some links. Let's go check out the next mnemonic. Alright, so now into mnemonic number 2. And what you're going to need to do on your scrap paper or in your mind is write out all of the receptors that are high yield, which are these: the alphas, the betas, those two together being the adrenergics, the muscarinics, or the cholinergics, whatever you want to call them, dopamine receptors, histamine receptors, and vasopressin receptors. So using the first mnemonic that I taught you, you could fill in each one of the GQ proteins that are associated with each of these receptors. But here's another way, just in case you like it better. And the mnemonic here is kiss and kick until you're sick of sex, which is written out like this. See that? Kiss and kick until you're sick of sex. And there you have it. Each receptor lined up with the type or the subtype of GQ protein that goes with it. And for your added viewing pleasure, here are a couple of short lists about what each of these receptors contribute to in our bodies physiologically. Okay, knowledge challenge number 1. A 16-year-old male presents to your emergency room. He's short of breath and wheezing. His mother's with him, and she's frantic. And she tells you that he recently lost his inhaler, but she can't remember what it was. However, because you are such an awesome medical student, you immediately identify this medical condition as asthma. And you realize that the inhaler was a beta-2 agonist used to cause bronchodilation. So, which G protein coupled receptor are we talking about? GS is the correct answer, so if that's what you picked, good job. Remember, you can write it out by either of the two mnemonics that we used. Knowledge challenge number 2. Which membrane-associated enzyme is the target of the GS protein? Well, that would be C, adenylyl cyclase. It also happens to be the associated enzyme with the GI protein, right? So what about protein kinase A? Well, protein kinase A is the final step in the cyclic AMP pathway. Any idea about protein kinase C? Well, that's one of the final steps in the IP3-DAG pathway. How about phospholipase C? Phospholipase C is the target of the GQ protein, and it is a membrane-bound enzyme that cleaves PIP2 into IP3 and DAG. All right, good job.